We left off talking about cell signaling in eukaryotes, and while this is part 2 of chapter 9, it will be quite brief. Part 1 is jam-packed with information, and so I wanted to leave some time for you to be able to go back and look at part 1 with the eukaryotic cell signaling, along with many of the websites I put into the module that have additional information for particularly the G protein coupled receptors, but also some receptor tyrosine kinase links. So our final slides for chapter 9 dealing with cell-cell signaling in unicellular organisms. And they do that primarily by a means called quorum sensing. Now, a quorum means you have a minimum number of participants. So many of the committees I participate on, we need a quorum of members in order to be able to vote on any actions. That means there has to be a specific percentage of the total membership present, otherwise the vote would not be valid. Now, unicellular organisms are not voting on academic practices, but they are sensing their environment and being able to respond as an entire population as opposed to individuals in that population to environmental conditions. So this is a link to a Khan Academy article. You can see the link pops up there when I highlight. There are a lot of resources on the Khan Academy. Toward the beginning of the semester, I sent out a link to the class I set up there that has resources for the various chapters, and I will send that out again. The quorum sensing in bacteria means they're using chemical signals, and those chemical signals allow them to detect the population density, because the more bacteria that are present in the population, the higher the concentration of the chemical signal. And the detection of that concentration of the chemical signal is what allows them to determine how many other bacteria are in that area. And then the behavior, the actions of all of the cells in that population will change in response to reaching the threshold of that signal. There really wasn't a good place to put me on here, so I'll just keep moving me around. So that threshold level depends on the signal, how concentrated, how many of those signal molecules have to be present in order to create that reaction. But then they will change their behavior or gene expression all at the same time. So now these unicellular organisms will behave very much like a school of fish that changes their direction all at once. So this might be movement or it might be gene expression. The production of a particular chemical in response to this threshold signal, or perhaps the repression of a chemical signal in response to the signal. The example that is best known for quorum sensing in prokaryotic organisms is in Alivibrio fischeri. It is an organism that is in a symbiotic, a mutualistic relationship with a squid. The organism, the bacteria, form colonies inside of the squid's light organ. The squid is not producing the light. The prokaryotic cells are going to produce light inside this organ, inside this animal. The symbiosis, the mutualistic relationship, comes from the fact that the squid provide food to the bacteria as well as protection inside the animal. And in return, these prokaryotic cells provide bioluminescence. And that's not what allows the squid to see. The squid has eyes, and the squid can see. It does provide additional light, but it's not the actual photosensing receptors. The bacteria emit light, and when that light is emitted, that prevents the squid from casting a shadow, because the cast shadow would be an indicator to predators that the squid is present. So without a shadow, the predators are not as easily able to detect the presence of their prey in the water. So how does this signal happen that's emitted from these prokaryotic cells that triggers a response in the entire population of those prokaryotic cells? There's a cute little squid. Move my image off of it so you can see it. We can see here the squid has its own eyes, but the light organs are located near there. Highly concentrated Vibrio fischeri in this region, so it is illuminated, but it's also in other regions on the squid. And that additional light is what prevents the squid from casting a shadow and protects it from predation. I got this from a National Geographic that shows not only are these bioluminescent bacteria providing protection from predation, but they also control the squid's circadian rhythm. That's why it says the glowing bacteria control the squid hosts. Our circadian rhythms, our wake and sleep cycle, are governed primarily by the presence or absence of light. So when it's daylight, we tend to be awake, and when it's nighttime, we tend to be asleep. There is dramatic variation in those cycles. My natural sleep cycle, if I don't have to set an alarm to go or be anywhere, is to be asleep around 1 a.m. That is long after it's been dark, no matter the season. And I wake up between 8 and 9 a.m. With work, even with teaching online, don't quite have that luxury. And so I get in bed and try to fall asleep earlier, and I wake up earlier. But these squid, their circadian rhythm is controlled by the populations of these Vibrio fischeri prokaryotic cells and their light organs. When the bacteria light up, that wakes up the squid, and when they are in lower numbers and decrease their illumination, the squid goes to sleep. This happens because of signals called autoinducers. If this was a multicellular organism, we had autocrine signaling. What a cell emits a signal that signals the cell that made the signal. The same thing is happening in prokaryotic cells, but we don't have any tissues. We are not connected to any other cells. So it's called an autoinducer because this cell has the genes in its DNA that will be transcribed to RNA and be translated to the other molecules. And in this case, we have this small chemical. When it gets secreted into the environment, if it re-enters the cell, the receptor is internal, and that would give it the signal that the autoinducer is present. And when it's low, those chemicals diffuse away. When there is a lot of the same cell present in a population, the autoinducer is in the solution away from it, but as it comes in, more of the receptors are bound by that inducer, and that regulates the transcription of the genes. So the genes responsible for the production of that signal get upregulated or turned on or transcribed in greater numbers, and so you get what's called a positive feedback loop. One cell, not very many autoinducers, there's really nowhere of the cell to receive it, not much happens. Many cells, each of them producing the autoinducers, the receptors bind to that autoinducer, That in turn binds to the DNA and upregulates or turns on the rate of transcription of those genes that make the autoinducer. So back to our little marine bacterium, Vibrio fischeri. This is controlled by a chemical called N-acyl homoserine lactone, or HSL. It interacts with a regulator, a protein called LUX-R. Lux is Latin for light, and that activates the transcription of the LUX operon at a high cell density. The operon is that series of genes. They all have a related function, and it is controlled by a single regulatory region. And so the LUX operon is a segment in the prokaryotic cell of genes that are all related to this production of bioluminescence. And this autoinducer interacts with this protein, the LUX-R, and that activates the transcription so that we get RNA copies of that gene, and we get the ribosomes to read that and make the enzymes necessary to produce that bioluminescence. And if you're interested in knowing more about this, I have provided the actual peer-reviewed journal article on this. Let's see if I can find a better place for me. This is a diagram of the LUX-R operon. This dark circle represents the bacterial cell membrane. This green double-ended arrow is representing the DNA, a very short segment of the DNA. And it's the DNA that contains these genes that will encode the information for the cell to make the chemicals that eventually result in light production. We have the LUX-R gene, the DNA sequence, that if it gets read to make RNA and translated to make protein, we create this LUX-R protein. That LUX-R protein binds to this light green area that is the promoter. The promoter is a DNA sequence where RNA polymerase binds. And that is the enzyme that is capable of binding to DNA and using the information in DNA to create an RNA copy. And we have to have that RNA copy for the ribosomes to read to make the proteins. So LUX-R protein binds to the promoter region. That allows the RNA polymerase to bind to the DNA, and then that will transcribe all of the other genes in the LUX operon. If they get transcribed, they will get translated. The first gene, LUX-I, is what will allow for the autoinducer to be made. That is the acyl homoserine that is going to get converted and make the SHL. They're going to diffuse out of the cell. If there's just this one cell making it, not much happens. But if many cells are making it, we get many of these autoinducers made that will come back into the cell and bind to the receptor that will keep this going and get light production. And again, I have the link to the article if you want to put this in greater context. This last slide is showing an image of a biofilm. On the left, that is a biofilm of Staphylococcus aureus that was formed on a catheter, so a tube inserted into a patient. This is not what you would want. These procedures are done very carefully so that the patient's skin is swabbed with an antiseptic to kill as many microbes on the surface as possible. The catheter is sterile upon insertion, but we can't completely eliminate all microbes in the air or on the skin of the patient. So if the patient had a low number of these Staph aureus bacteria that got onto the surface of the catheter, when the catheter is inserted, we now have a warm, moist, nutrient-rich environment inside the patient for those bacteria to grow and replicate. And when they are in close contact, again, once there are enough of them, they secrete autoinducers that indicate the level of population has reached a threshold, and then they trigger the entire population of the Staph aureus to secrete toxins. The toxins cause great disease in the host. If they do it for their own benefit, for the bacterial benefit, they will make the host sick enough that the bacteria are excreted through blood or wounds or body fluids, but not kill the patient. However, Staph aureus infections from this sort of situation with biofilms can actually lead to the secretion of enough toxin to cause grave illness and death. And then this is just another picture of our cute little bobtail squid who is glowing because of its symbiotic relationship with enough Vibrio fischeri in its light organ to cause the bioluminescence. That wraps up Chapter 9. Today's lecture extremely brief, but the previous one certainly was not. We will be moving in to new topics after this. Chapter 10 will happen next week, and so keep track of your lecture discussions. The lab notebook should be up to date at this point, and I will make sure that the lab discussion forum is also available.